Hi, I'm Li Ma, a professor of biochemistry and molecular biology at UMass Amherst. Welcome to the MALab. Our research focuses on Fusarium oxysporum, a fungus found everywhere. However, when it does not behave itself, it can cause disease on important crops and infecting humans. For instance, it's a cause of Panama disease of banana, and you may even see it in your backyard wilting your garden tomatoes. It can infect immunocompromised patients, and it's responsible for the contact lenses-related keratitis outbreak. Such bad behavior qualified this fungus as a cross-kingdom pathogen. Empowered by supercomputing capacity through the Massachusetts Green High Performance Computing Center, we have demonstrated that what makes this fungus bad is the accessory chromosomes, which carry unique genetic elements to target specific hosts. My group's composed of many talented young scientists, including postdoc, graduate students, and undergraduates. We are working together to better understand this unique pathway system for the effective control of this cross-kingdom pathogen.
Hi, I'm Vista Saurabh, an undergraduate researcher in the MALab. One of the topics actively studied in our lab is the genome evolution of Fusarium oxysporum strains and the resulting effect on host pathogen interactions. In addition to mutations, transposons also contribute to genetic variation. These mobile elements are repetitive sequences, also known as jumping genes, that lead to changes in genome architecture and gene expression. To identify such insertion events, we developed a simple pipeline using the high-performance computing cluster that incorporates common variant calling software to report transposon insertions absent in the reference genome. The resulting output files are visualized here along with the alignment file. The colored and split reads are indicative of a true insertion event at this genomic location.
Hello, my name is Dilay Ayhan and I am a grad student in MALab. My project focuses genome evolution of cross-kingdom pathogenic fungus Fusarium oxysporum. I used the resources at MGHPCC to analyze genome and transcriptome sequencing data to find out the methods of adaptation in different Oxysporum strains.
Hello, my name is Ho Lin. I am a graduate student of MALab in plant biology program. My research topic is plant pathogen interactions using arabidopsis and Fusarium oxysporum as a model system. I have been taking computational approaches such as transcriptomics and comparative genomics. Here I am showing a protein-protein interaction network computed at MGHPCC, reflecting how plants respond to the fungal stimuli.
Hi, I'm Jacob Maman.
And I'm Kelly Allen.
And we are graduate students in the MALab.
We study agricultural diseases of sweet basil caused by the fungal pathogen Fusarium oxysporum and the oomycete pathogen Peronospora bulgarii.
We utilize MGHPCC to analyze genomic and metatranscriptomic data in order to identify genes and processes involved in basal disease resistance to pathogens and to understand pathogen virulence strategies.
One project that we are working on is identifying and testing basal genes that can provide resistance to basal downy mildew, a devastating disease that causes millions of dollars of agricultural crop loss each year. Our lab identified several candidate resistance genes using the MGHPCC to analyze transcriptomic sequencing data from a comparative study. And now we are working on characterizing our top candidate gene so that plant breeders can use it in molecular marker-assisted breeding to produce healthier crops.
Another focus of our research is identifying and characterizing pathogen genes involved in infection. We predicted these genes called effector genes from transcriptomic sequencing data using MGHPCC to identify expression profiles, conserve protein structures, and sequence motifs. We use this data to design our experiments to test our hypotheses about how these pathogen effectors influence disease outcome in agricultural crops.